Hello everyone, welcome to this new seminary of the same series. So today we have Dr. F Paschalidou presenting her research, and I would like Magali Profi introduce today's. Thank you very much.
So good morning everyone, it is a great pleasure to welcome Fotini. So Fotini, she has developed interdisciplinary research. She's combining clinical ecology, plant biology, and insect behavior. So since she left Greece in 2006, she has built an international career with several leading European research institutes. So, for example, she has been working in Wageningen University and in Netherlands, also in ETH Zurich in Switzerland. So her research seeks to understand how plants perceive and respond to information for their environment, particularly insect-derived cues, plant signaling molecules, and volatile organic compounds. She investigated how these different cues influence interaction among herbivores, pollinators, natural enemies, and plants, and how these different processes are shaping all the multitrophic levels around the plant. So she has also contributed in a sophisticated way. Plants gather information and adjust their defense and interaction with other organisms. So a major recognition of the originality of her work and the impact of her work came since 2025, where she got ERC starting grant for the project Panoply, that she's going to present some general objective of these different projects. And she has been previously working in the agronomic unit already in Paris, and she's now working in APSIS research unit in Montpellier. So I'm not going to tell too much about the Panoply project because she's going to present the different objectives, and now for Tini, the floor is yours, and we have your table.
Thank you, everyone, for being here today. Thank you, Magali, for this very nice introduction. And so, as Magali mentioned, today I'm going to present my work on plant cues and their effects on multitrophic interactions. And I would like to take this approach and this concept to agroecology. But first of all, because insect pest outbreaks can cause an economic and agricultural devastation, such as the phylloxera example, that destroyed the 2/3 of the European grape vines in the 19th century. Of course, since then, we have advanced in our knowledge regarding pest outbreaks with pesticides, which of course raise environmental and health concerns, biocontrol, which is restricted to certain species interactions, and by genetic engineering that takes long to be used. Therefore, insects still remain a major agricultural challenge. And one potential avenue for sustainable pest management lies on understanding chemically mediated multitrophic interactions. And what I mean here with multitrophic interactions is the sum not only of the direct interactions between the plant, the herbivore, or the herbivore and the natural organism, but included as well indirect interactions, such as plant-plant interactions, or pollinator-natural enemies interactions, or pollinator-herbivore interactions. So, one factor among others that mediates these multitrophic interactions is the flow of chemical information. And volatile organic compounds, so compounds that are emitted from the plant to the environment, can affect the behavior and the physiology of all the organisms in the multitrophic interactions. And to explain a little bit more about volatile organic compounds, I will introduce you the most well-known volatile compounds that have an effect on and mediate trophic interactions, which is the herbivore-induced plant volatiles. And now I want to show you a little bit of a video for the ones that they are not familiar with the herbivore-induced plant volatiles, to explain a little bit how they can mediate trophic interactions. I have already shown this video to my daughter, and she liked it. I hope you like it too. So here you see a plant, it's intact. So the plant emits constitutive volatiles, these green things that come off of the plant. When there is herbivory, so when there are herbivores feeding on the plant, you'll see that the volatiles are changing. So it means that the profile of the volatiles of the plant is changing, either with production of new compounds or with the changes of the ratio. Natural enemies like parasitic wasps use these changes of the blends and try to locate their hosts. When they arrive on the plant, they use short distance cues, such as volatiles, short distance volatiles, or the phrases of the herbivore. And when they locate the herbivore, this is a parasitic wasp, they lay eggs inside the caterpillar. The caterpillar does not develop, and then it dies. So this way, the plant is protected by calling the body guards. So this is the most famous example of a chemical compound that can mediate trophic interactions. Yeah. So, as I said, parasitoids or natural enemies use herbivore-induced plant volatiles as a long-distance cue to locate the hosts. They can be produced by the plants in large amounts. Some of these compounds can be produced de novo, some they change the ratio. And what is very important and interesting is that these volatile profiles of the HIPVs can be herbivore specific. So in other words, here you see a parasitic wasp laying eggs inside the larvae of a caterpillar. So the plant produces specific HIPVs to attract these specific species of the parasitic wasp. And here you see another parasitic wasp, another species that attacks aphids. So this plant, which is attacked by the aphids, produces a specific blend to attract the parasitic wasp for the aphids. So these all are very interesting and very exciting, but also these HIPVs, except from mediating trophic interactions, they can mediate plant-plant communication or plant-plant interactions. So when the plant is attacked by a herbivore and emits herbivore-induced plant volatiles, the neighboring plants can recognize these HIPVs as a cue for future herbivore attack. Because as you can imagine, the herbivores, when they feed on this plant, they can distress and damage their neighboring plants as well. So the plants respond to these HIPVs with different ways, with different types of defenses. One can be induction and one can be priming. So induction is an immediate response to stress, but I'm not going to discuss induction in this presentation today because I'm very much interested on priming. So priming is that the plants are on a state of alert. So when this plant is attacked by the herbivore and emits the volatiles, the other plant, the neighboring plant that can perceive these volatiles, gets ready to battle the herbivore when the herbivore arrives. So it gets to a state of alert, and this enables faster and stronger plant defenses when the herbivore arrives. So the main question is, if we can prime the plants with chemical cues, can we prime crops on purpose to increase the defense and increase the resistance against herbivores? And if yes, how priming might affect multitrophic interactions in agroecology? So these are the main big questions during also the period of my career that I was interested in. And I started working with Brassicacea species, where I wanted more to understand how different cues, such as volatile organic compounds, or the presence of eggs of the herbivores or chemical elicitors can mediate multitrophic interactions from the laboratory to the field. So my initial question was related with the eggs of the herbivore. So I wanted to understand whether plants can use eggs of the butterflies, of the herbivore, as an indicator for future herbivore attacks, because the eggs will hatch, the caterpillars will start feeding on the plant. So I wanted to see, in other words, if the plants are primed because of the presence of the eggs. And here you see Pyrrhus brassica, a large cabbage white butterfly, laying eggs on a Brassica nigra plant, a cluster of eggs. This is how these eggs look like under the microscope, super cute. And this is the caterpillars that they come out and then they start feeding. So the first question was how egg deposition by different herbivores can trigger prime responses and can trigger priming in Brassica nigra. So I used two different herbivore species. One is a specialist on Brassicacea. Mamestra is not a specialist, can feed on everything. And then I wanted to check as well the parasitoids in the lab and in the field. And what we found was that indeed plants can recognize the eggs of the specialist, not the generalist, prime their defenses, defend stronger against the herbivore. And that's what we also find that was interesting and not expected, was that the plants, when they perceive the eggs of the specialist, they start shifting to reproduction faster. So they recognize the eggs and then they start producing flower faster, and also they produce faster seeds and in higher numbers to avoid the herbivore. This is what reproductive escape means, because these herbivores are feeding on the flowers at a certain point. So for the plant to safeguard the reproduction, they shift faster to flowers, and the herbivores do not feed on the flowers anymore because they are already seeds. And that happens only with the specialists. And in the field, what we also saw that plants that they were primed by deposition had a higher number of natural attraction, and the parasitoids had an increased performance. So then after that, I moved to Zurich in '88 for my postdoc, but I was still interested to understand more about these early warning cues for future herbivore attacks, such as the eggs. And as I mentioned before, we already knew at that point that herbivore-induced plant volatiles can prime plants that they are in their vicinity. So I was interested to see whether we can prime neighboring plants by oviposition-induced plant volatiles. So we had our plants that emit oviposition-induced plant volatiles when Pyrrhus brassica lays their eggs, and I wanted to see whether the receiver of these OIPs can be primed and what is the effect on the herbivore. So we used different Brassicacea species where we identify certain cues from the oviposition-induced plant volatile plant that can prime the neighboring plant. We found reduced herbivore performance, and we also saw the same thing that we have seen on this species when we had the eggs. So the receiver of the oviposition-induced plant volatiles shifts to reproduction, induces flowering and shifts to reproduction, except from the priming of candidates. So the plants, by detecting oviposition cues, either the eggs themselves of the herbivore or the oviposition-induced plant volatiles, they prime and they defend faster and stronger, but they also prioritize their reproduction to maximize overall fitness. That was very interesting. But in the experiment, we found something that was much more than this, and now I can share an anecdote during my postdoc, what happened there with the experiments. So in order to measure the fitness of the Brassica nigra plants, we used bumblebees to pollinate our plants. And then, when we finished with the experiment, we put the bumblebees in a growth cell together with Brassica nigra plants that did not have any flowers. It was on a vegetative state, and we were going to use them for another experiment. So we left the bumblebees there with the plants, and then we continue our experiments. Well, the bumblebees were actually starving for pollen, and then at a certain point, one of the students came fast to our office, and they said, you have to come down because the bumblebees are attacking the plant. The bumblebees usually pollinate the plants, they don't really attack the plants. And then the student was insisting, no, you have to come and see. They are actually damaging the leaves of the plant. So we went, and indeed, here you'll see, the bumblebees are damaging, making holes on a vegetative plant, a plant that did not have any flower yet. So we thought, okay, that might be an artifact. So we tried to make photos and understand how is this damage coming from. And indeed, the bumblebees were damaging the plants. We also tried to see whether there is any parts of this plant species that they take back to the nest, maybe for feeding or we don't know for what. We couldn't find any parts of the plants on the nests. And then we used some other also different kind of beehives from different companies. And then we kept saying keep seeing the same thing. So we start thinking about the annual cycle of the bumblebees, and then thinking about when the queen overwinters, she starts the nest, and then very early in the summer, the bumblebees need a lot of pollen to establish the colony. If there is not a lot of pollen in the beginning of the summer, the colony can die. So they cannot develop it anymore. So thinking that in our experiment, completely by chance, we had started the bees with pollen, we start thinking that whether the bumblebees may actually be able to influence their local flower availability by damaging the plants on the vegetative state, so the plants can accelerate flowers, the flower production. So we wanted to understand whether the bumblebees accelerate flowering in two distinct species. And to test this, we used tomato and black Brassica nigra, the black master. And what we did is we let the bumblebees damage a number of plants. Then we tried to mimic the 5 or 10 holes maximum with a forceps in a mechanically damaged plant. And then we had a control which was completely undamaged. And then we measured the first flower appearance in the tomato and the first appearance of the flower buds on the Brassica nigra. And what we found was that indeed, that the plants that were damaged by the bees flowering significantly faster compared with the mechanically damaged and the control for the tomato and also for the Brassica nigra. And in total in tomato, we found that the bee damaged plants were flowering 30 days earlier and 25 earlier according to mechanically and control. And for Brassica nigra, that was 16 and 8 days respectively. So indeed, we saw in two species that this bee damage can accelerate flower production. But then we were thinking, is this related with the pollen availability? First of all, due to the annual cycle of the bees, and second of all, because we knew that our bees, when they first showed this behavior, they were starving. So we wanted to test this, and to do that, we have two distinct hive treatments. One was satiated pollen, and they could eat as much pollen as they had. Then they were able to damage the plants, and then we reversed the diet. We gave them pollen-deprived diet, so they didn't have any pollen, and again the opportunity to damage the plants. And on the other hand, we reverse. So we have not giving them any pollen, give them the opportunity to damage the plants, then giving them pollen-satiated diet and the opportunity again to damage the plants. And we saw that indeed the bumblebees that they were pollen deprived were damaging the plants much more compared with the bees that they were pollen satiated. So, and that was regardless in the order of which the treatment was presented. And here I have to say you also something, an anecdote again, that we actually contact certain companies that they were having these bees and we got them from them. And then we start mentioning this, like, have you seen your bumblebees damaged? Have you heard back from someone saying that the bumblebees are damaging their plants? And actually they told us yes, that they kind of knew this phenomenon. And that's why they always provide pollen. And that's why they always say, give them a lot of pollen. Because actually, some people from the company, they are not interested, of course, why and how for the behavior. But we were not actually the first ones that saw. They were people already knew. But of course, yeah, we have different objectives. So, as many other studies, we have more questions than answers. And of course, the two most important questions related with this damage is how might this behavior has arisen for the bumblebees? One hypothesis might be learning, because of course we know that bumblebees are amazing creatures learning and they have great skills and abilities. But I have to say here that this is not the strongest hypothesis, because even for these amazing creatures, we can think that if they damage a plant, it will take quite some time until the plant is flowering. So it might be very difficult for the bumblebees to come back exactly on the same plant that they damaged and remember it for getting future reward. So yeah. The second hypothesis is that there was an immediate reward of the plant, either which is related with the fitness of the bumblebees, either a plant sap that we were not able to measure, or some chemical information that we were not able to identify at that point. And the third hypothesis is that maybe this behavior of the damage is related with the nectar aerobic, because the bumblebees are able to get nectar from flowers. However, might be this, so the bee damage behavior might be an adaptation from the nectar aerobic behavior. And also, another burning question, still all of these questions are a bit open. We know some things more now from the lab at TTH, but still they are very open. Why an adequate response to this bee-driven leaf damage would accelerate flower development? So again, we see here there are three main hypotheses. The one is that the plants perceive the damage by the bees as a herbivore attack, and they shift to reproduction, something that we saw with the presence of the eggs or with the neighboring plants that they receive the chemical cues. The second is that there is an adaptation by the plants. So the plants, when they recognize the bee damage, they start flowering faster in order to overcompete other plants for the excess of pollen in their vicinity, among other species. And the third one is that this has nothing to do with the plants. That might be a chemical manipulation, a chemical cue, a volatile, or something that we have not yet identified, that manipulates the plants, and the plants flower faster by the bees. So these are all still open questions. And coming back to the flow of the chemical information, I would like to highlight that yes, we have some knowledge for certain cues that they can affect, mediate multitrophic interactions, such as the HIPVs, the volatiles, or the OIPVs. But still, there are so many things that we don't know that can mediate multitrophic interactions and worth to investigate. So when I moved here to France, I wanted to continue working on the same kind of topic. And I was granted with a JC starting grant, which was about understanding how again, the oviposition-induced plant volatiles mediate plant-plant communication and how this shapes multitrophic interactions in agroecological plants. And this is actually now the work that I'm going to show you of my brilliant PhD student, Pius Otto, that he defended his thesis last December, and Alain Pertintoy, our collaborator. So we used a new species, Brassica napus, which is related with Brassica nigra, but still we had to establish whether there is OIPV priming of neighboring plants. So what we did was that we had the butterflies laying eggs, the plants were emitting the OIPV-induced plant volatiles, and we wanted to understand whether the receiver is able to be primed. And now I'm going to explain you a little bit more also on the methodology that we had developed. So we had two treatments. The first was the priming treatment, and the second is the control treatment. So we let the butterflies lay eggs on the plants. The plants emit OIPVs, and then this will have a plant which is the receiver of the OIPVs. And the control is an intact plant that emits constitutive volatiles, and then we have the control of the receiver. So we exposed the OIPV receiver and the control receiver to the OIPVs and the constitutive for 4 days. And then we collect the volatiles of this at this point of the different treatments. After 4 days, the caterpillars are hatching from the eggs. And we have the OIPV emitter with caterpillars. We transfer a number of caterpillars to the OIPV receiver. And we also have the number of caterpillars of the control receiver. Then we collect the volatiles of 3 and 7 days of larval feeding of the total treatments. And we collect also, and we measure the larval biomass to have an indication of the prime. And here I want to show you the step by step some of our results. So what we found was that the profile of the OIPV emitter, which is this, the red, overlaps with the profile of the OIPV receiver. And here I want to remind you that this OIPV receiver plant is just the neighbor of the plant that emits the volatiles. So if you see the neighbor at a certain level copy pastes the volatiles of the OIPV emitter of the actually infested plant. And the control is separate. Of course, when we have feeding, we have very different profiles of the volatiles. But you still see here the overlap of the OIPV receiver, the OIPV emitter with caterpillars. And this control now that has caterpillars, we see that there is a similarity in the profiles. This yellow plant is a control emitter which is intact, so it's normal that it's separated because it does not have any eggs or caterpillars. And here, 7 days after feeding, because we have the herbivore-induced plant volatiles, we see a total overlap of the profiles for the plants that they have the caterpillars. And of course, the intact plant is very different. So we wanted to better understand which are the cues that can prime the neighboring plant. So which are the responsible compounds for the priming of on-plant-plant communication? And at that point, we were able to identify 3 compounds, alpha-pionine, dimethyltripsylphane, and lamin, which were much higher elevated on the plants that they had the eggs. And these are different compared to the rest of the treatments. So, and I have to highlight as well here that these compounds are quite well known for their effect on trophic interactions, attraction of natural enemies, and so on. And then we wanted to check for priming of plant defenses, and one way to do that was to measure the biomass of the herbivore. So the caterpillars that fed and developed on plants previously infested with eggs, they have lower biomass. Also the caterpillars that developed on the receiver of the OIPVs, compared with the control for 3 and 7 days. So at that point, we wanted to take what we have found in the lab in the field and see in the field we can also see priming of plant defenses by OIPVs and also the effects on different insects. So we had, okay, back. So we have a treatment where we had control volatiles, which is the solvent. We have a treatment that we had the natural OIPV emitter and the synthetic OIPV. So the three compounds that we had identified. And then we wanted to measure defense, growth, and reproduction in the field. Here I have to say that we did these experiments during June in Paris region, and still the weather can be very bad. So the butterflies did not want to lay any eggs. They almost freezing to death. So we had to cancel the part with the natural OIPVs, unfortunately. Luckily, we had identified synthetic OIPVs that could work, and it worked out. And so we continued with the synthetic OIPV treatment and the extension. And what we did was we refilled the dispenser for 5 days, the dispenser of the synthetic OIPVs and the dispenser of the control of the volatiles to mimic the natural oviposition time that it takes 5 days. And then we stopped. We did not fill the dispenser at all after these 5 days. Then we put caterpillars and we measured the weight of the synthetic OIPV receiver and of the control receiver. And we also measured different kinds of proxies for growth and reproduction. And what we found from the field was that the caterpillars that developed on the synthetic OIPV receiver had much lower biomass compared to the control. And we did not find in this case any difference on the growth and any difference on the reproduction. So this was a bit controversial to our previous results when we had the OIPVs in Brassica nigra and the plants shifted to reproduction. In this case, we did not find that in this experiment. There are many reasons can be why. First of all, maybe there is no effect on their growth and reproduction. Maybe the weather that was completely very bad did not allow the plants to grow well because we had anyway very low numbers of seeds, even on the controls. So yeah, there are many ways why this did not work in the field. But the other part that was interesting and we wanted to develop was how does exposure to the OIPVs save the insect community dynamics in the Brassica nanosphere. So on the same plots where we had the synthetic OIPVs and the controls, we measured the different insects. And in more detail, we collected the insects on yellow traps and personal observations. And we also identified the insects on families and functional rows. And here I want to remind you that we had exposed the synthetic OIPV receivers and the controls to the control volatiles on the synthetic 5 days, and then we stopped. And then we measured the insects up to 2 weeks after the exposure. And when we checked the within plot and among plot diversity on the plants that were the controls, exposed to the controls or exposed to synthetic OIPVs, we did not find any difference. So that means that we have the same species on the plants that were exposed to the OIPVs and the same species and families on the plants that were the controls. But when we start looking a little bit more on the functional groups, we found that the plants that were exposed to the OIPVs had higher number of parasitoids and predators and pollinators compared to the plants that were not exposed. That's not the same for herbivores and other insects. But here, again, I want to highlight that these plants were exposed only 5 days to the OIPVs. And 2 weeks after, we could see that still they had more predators and more pollinators attract them. And then going a little bit further on the families, we found that Ichnomonidae families, which are usually the parasitic wasps and the predators of parasitoids, and Apoidae families were increased significantly on the plants that they were exposed to the OIPVs compared with the control. And we saw a decrease in aphids. So the aphids is another herbivore on the plants. So the plants that they were exposed to the OIPVs had much higher numbers of parasitoids and pollinators, and less number on aphids, which is very interesting, I think, results. So overall, to close this very nice chapter of bios, we found that the emitters of the OIPVs, more specifically the compounds alpha-pionein, limonene, and dimethyl-5, can prime neighboring plants in the lab and in the field, does not have any differences on the plant fitness, can have an effect on chewing herbivores such as the larval biomass performance, can have a reduced number of aphids, and can increase the attraction of subfamilies of parasitoids and bees. And at that point, I had to move from Paris to Montpellier, and then I had to explore what I can do here and how can I work here. So I visited multiple labs, and then when I was saying all the nice stories for me about the OIPVs and priming and on Brassica cannabis, nobody was interested in Montpellier because nobody works with oilseed rape here in the south. However, everybody was interested on wine and grape wines. So I had to develop a new project on grape wines, and I was fortunate to receive an ERC starting grant, as Magali mentioned, on this new system, on Panoply, which still wants to investigate how plant volatiles mediate species interactions. And Panoply in Greek means a complete suit of armor. So we want to understand the comprehensive defense system of the plants in relation with the volatiles. So, as I said, I changed the system. So now I'm working with grape wine, which is relevant agriculturally and economically. I work with the leaf hopper Scaphoideus titanus, which is able to transmit flavescens doré. Bear in mind, there is no alternative except the pesticides for Scaphoideus, and there is no treatment for flavescens doré. So the farmers here in France and everywhere, if the plants are affected with flavescens doré, they cut them and they bury them. So that's why also as well it's interesting to think alternative solutions for this kind of organism. And then we want to include the aromatic plant as the source of volatile organic compounds for many reasons. First of all, because they have the potential in studies in other systems to reduce the pest and attract the natural enemies, but also many of these volatile organic compounds may act as a priming cue for the receiver. And this is why I'm very much interested as well, whether in this project, whether there is a priming effect that can mediate this trophic interaction. So to answer these questions, we have these different experimental approaches and systems. First, we want to start with the plants to see how do grapevine plants respond to the box from aromatic plants, and more specifically from thyme. This is together with Lorraine, our postdoc, and Magali, sits here. Then we want to better understand how and which box affect antagonistic interactions from the lab. And this is the work of another brilliant PhD student, Salome, who is also here, together with Nicolas Sagnon. Then we want to better understand how and which box in semi-natural conditions or microcosmos, mesocosmo experiments can affect mutualistic interactions between plants, natural enemies, and pollinators. This is in collaboration, with Enrique Frago from CETESP. And last but not least, we want to better understand which is the overall effect of box in multitrophic interactions in the field. This is in collaboration as well with Elena Kazaki from the CEF. So this is how I would like to come to the end also to my presentation. And this project aims to give like a new insight in chemical ecology, which is a very relatively new domain on agroecology, but in general as well. Understand ecological mechanisms in the field. Also, how by understanding these ecological mechanisms in agroecological field, we can safeguard ecological services such as pollinators and natural enemies. And we aim to be able to transfer this idea of box system to manage insects, and that can be also disease vectors, and set the foundation for novel strategies towards modern sustainable agriculture. And this is where I would like to close my presentation. Thank you for your attention, and I'm very happy to receive any questions.
Thank you very much for the talk. So don't hesitate if you have questions. And online, you can put your question on the Q&A or directly raise your hand. I will give you the floor.
I have a question about the bumblebee experience. You said you had 3 hypotheses here. I'm not sure I see the nuance between the third hypothesis and the other hypothesis.
Which one? From the nectar robbing, you mean, or?
No, sorry. The third hypothesis, it was a way of
The chemical manipulation. So, for example, we have seen that in other systems, not bumblebees per se, but in insects, that viruses can manipulate insects to change the behavior. Or the viruses can manipulate the plants to defend in a different way or not defend. So this is related more with how. So this is not an adaptation. It's like it has the fitness benefit only on the bumblebees, because even though we saw the induced flowering, we don't know, we did not measure if there is a fitness benefit for the plant at that point.
So is there benefits for plants when they flower earlier?
So this depends on the cue. For example, when we have the OIPVs, when the plants receive the OIPVs or the eggs, they flower faster, but they have also higher number of seeds. So this has an increased fitness benefit for the plant. But in this case, we have not measured that. So we cannot say for sure that there is an adaptation if there is not a fitness benefit for the plant and for the bee. So as long as we don't know if there is a fitness benefit for the plant, we cannot say that it's an adaptation. It might be a manipulation by the bee. Does this answer your question?
I guess I'm still like a little, like, it's clarifying a lot of it, but
Okay.
Yeah,
It's okay.
yeah.
If I may follow on this, just so if I understood, it still could be, if the bumblebees are manipulating, they could be manipulating an adaptation of the plants to the herbivores. So that's why it can be overlapping view. That's
Yes.
what I understood.
Yes. So the thing that I know a little bit more how the science has progressed in this case. So I know that they have kind of identify the cue that can induce the flowering in the bumblebees, and it's not the same with the herbivores. And I think it was.
I didn't understand the second hypothesis. How could the plants? Why could the plants adapt to this?
Yeah. So the idea there is that if the plants can receive the bee damage as a cue of the bees themselves, they can start produce flower faster. So the plants that they have this adaptation, they can outcompete for pollen the rest of the plants, because they will be the first ones that they will flower. So the bumblebees will, and not only the bumblebees, other species as well, they find these plants first. So the first that will flower have higher chances of getting pollinated. So yeah, but I know still also we try to understand why and how and yes.
Yes, also on the table topic. How do you say that the bumblebees are not getting anything to the nest, no benefit? How do you know? Maybe there is something on the leaves that it's I don't know, somehow useful for the development of the larvae or for other things in the colony.
So we dissected, we did the dissections on the hive, and then we also dissected the larvae and analyzed, and we saw that there is no something that they transfer from the plant to the hive. So this is how we say that probably there is nothing that they can transport to the hive. But we might miss something. But I think that they don't, because the other thing that we've done was an experiment where we had the plants, and below the plants we had like a paper cone, and we could collect all the pieces of the plant. So they were cutting the leaves, and we were able to collect all the small pieces of the leaves.
Yeah, but maybe there is something like some
Tiny.
Yeah,
I know.
This
we cannot say 100%, but we can say very high probability that they don't transfer. I don't know.
I had one question. It's maybe a methodological question, but when I saw your experiment on starving and non-starving bumblebees, my first idea was you do starving and non-starving, but you did more than that. You did. What's the methodological? What's the advantage of this approach?
It's more or less because we wanted to see whether we reverse. If we reverse, so for example, if they are just starved, and we wanted to reverse the treatment to give them pollen to see whether they will continue or they will stop damaging the bees, the plants. So it was more like a prior experience. If they have a prior experience of pollen starvation or of pollen saturation, would they change their behavior later or not?
To me, my guess would be to have two bumblebees different, and one experiment is they are starving and the others are not starving. From that, you already can conclude that there is an effect.
Yes, indeed, you can conclude. But then we wanted just to see whether we can reverse it or not. Yes, but yeah, indeed, we could have stopped it there. Is there someone also? Okay.
Thank you for that. I was wondering if you have some experiment about like the maximum distance to which one insect can detect a volatile. Like I can get like in an ecosystem you have like spatial patterns.
Yes, yes. So I have not done that, but there are other colleagues in the field that they have tested at least until 60 centimeters. It has been shown that the OAPPs can be received from the plants, but then when we talk about the insects, it's much larger. It can be meters. For sure, for the parasitoids, we know that they can detect for very long distances the differences in the volatiles.
And then could the plants prime the other ones?
That's a very good question.
No.
We don't know. This is a very good question. We don't know. Also, then it's related as well, I guess, with kin recognition. So if the species, we have the same species, the one can prime the other due to kin recognition, that can be a mechanism that can.
Yes, so following up on this multitrophic interaction, so for the bumblebees, but also for the larvae of the butterflies. From what I remember, there's something in the saliva also of the insect that induces change.
Yes.
So can you tell us more about the specificity of the response of the plant?
So, like I said, I don't know the whole story, but I know that the plant responds differently on the saliva of the herbivore compared with the epidermis.
And between the different herbivores?
Within the different herbivores, if we have different
herbivores, if you have the different herbivores, let's say, do you see different herbivores?
Yes.
Maybe on the plants that you have been looking.
Yes.
Did you only try with one? No, you tried with different.
With Mamestra as well, and
yes.
it did not work. We did not see any priming by oviposition or something like that. But also we know, for example, that the aphids when they are damaging the plants, they have different HIPV profiles than the caterpillars, very distinct. So how these can prime neighboring plants, I don't know exactly, but my guess is that some compounds overlap. So I guess that might be some more general volatile cues that plants can use that are related with insect herbivory. And of course, the parasitoids is another story because the parasitoids have evolved to detect very specific changes. So
I've got a question on this project. I was wondering, is there an interest also of looking at real vines as a emitter also, or no?
yes, so it is very interesting to check whether the grapevine, when it's damaged by Scaphoideus titanus, for example, can prime the neighboring same species. And this is part of another project that it has been deposed, but I don't know it's going to be funded.
So complementary to this one.
Yeah, it's larger. But if, of course, it's very interesting.
There are no more questions. I don't see any question online. I would like to thank you again for this.
Thank you for being here.
Just before finishing, for people here and also online, I just remind that the call for the next semester speakers is still open until Sunday. So do not hesitate to post some. Thank you very much.
Yes, thank you.